Okay, we're going to move on, if we may. We're going to come to disitamab vedotin. Disitamab vedotin is an agent that as monotherapy in HER2, 1, 2, or 3 plus, which is about 70% of patients with a response rate of 50%. It varies a bit, again, depending on the data cut. It looks to have less skin toxicity, but it does appear to have fatigue, and it does appear to have peripheral neuropathy. It looks like an active agent. Matgalski presented DV Pembro data with 75% frontline response rate. And in our previous session, we described that neoadjuvant cohort with a 61% path CR rate, with all of the caveats of the neoadjuvant assessments that we talked about in our previous lecture. What do we make of this drug, Andrea, and where do we go with it? It is in randomized phase 3. It's essentially repeating the EV-302 trial with that same control arm, which is going to be difficult in America, Germany, and France, where EV Pembro's approved. What do you do with EV? What do you do with DV? It looks like the biggest victim of enfortumab vedotin's success is disitamab vedotin. Had we not had this, would have been out of the world data. But coming back to what Petros said before, I suspect there's cross-resistance with the same payload. We don't know that yet. So where does this drug land?
It's a great question, and I think this data is super exciting, but it is the same payload. So, and you have to have the biomarker, but what we saw in the most recent data presented by Matgalski at ESMO was that you can be HER2 low. And if you count the HER2 lows and the HER2 highs, we're talking about 80% of the patients with bladder cancer. So now we're not talking anymore about a really small number of patients. We're talking about a much larger number of bladder cancer patients. So where would it fit? I mean, the response rate over 70% when you combine it with a checkpoint inhibitor, so I think that's really exciting. It seems to have activity in the neoadjuvant setting. There was another study, I think it's this one that was presented, that has longer term follow-up in combination with a checkpoint inhibitor that again has an overall survival of 32 months. That was a poster at ESMO. So I think that this drug is very active. We need to figure out where is it going to fit. Is it going to be toxicity? Maybe patients that have rashes or have hyperglycemia. This could be a potential option for those patients in the same setting that you would use EV plus Pembro. Potentially, we need to see the bigger data set, but I think this is an exciting molecule.
I'm going to ask the four of you whether you think there's going to be cross-resistance between EV and DV.
I think there is. I think when you use the same payload,
yes.
I mean, it's like kind of using the same chemo, but the nice thing is that they do have different targets. So potentially you can avoid certain toxicities that are associated with the target and not the payload. But I think with the payload, they're probably going to have the overlapping in terms of the efficacy. It's going to be very similar, but I think that we need to study that prospectively. Like can you, I don't know, this is crazy, but can you potentially give both and see, you're attacking, the payload with two different targets. Is that something that's feasible? Are you going to have higher responses or higher toxicity? There's a lot of questions we don't know, and it all goes back to what are the mechanisms of resistance of these antibody drug conjugates, which we know is not only in the payload, but it's also on the target. It has to do with the linker, with the efflux of the drug. So I think there's a lot of different mechanisms of resistance.
We're going to keep going. We're going to move on from disitamab vedotin, and we're going to move to TDXD, which is also a HER2 ADC, but it has the potential advantage of a different payload, assuming that EV Pembro is going to be widely used, or EV Durva-Tremi potentially in the neoadjuvant setting. TDXD is an agent that has FDA approval, and you're using it in the US. I've used a tiny amount, and I haven't got a huge amount of experience. One or two private patients come darken my door with having been on an array of drugs. But I speak to my colleagues and friends in the US who quite like this agent. The label is associated only in the HER3+, which is only 20% of patients, not the 70 or 80% that Andrea mentioned. When you look under the bonnet at this basket study in detail, that only represents 16 patients. It's unusual to get an FDA approval of only 16 patients. The response rate is 52%, which is high. But when you look at the concordance of the immunohistochemistry done centrally and locally, the concordance is lacking. It actually looks like we're not good at measuring HER3 anyway. So what's high to one person seems to be different from what's high to another person. This therefore appears to me to be a hugely grey area around defining this high population, but this does look like an active drug. Is this more promising than trastuzumab govitecan? Or is this going to end up in the same basket with nice phase 2 data, accelerated approval, but a negative randomized phase 3? What's the future of TDXD in bladder cancer? And Shilpa, I'd like you to start with that.
Yeah, I think we can't really compare it to trastuzumab govitecan. That is not a biomarker selected drug. This one is for HER2 3+, and to your point about the IHC, this one follows a unique measurement of the HER2 based on the gastric cancer, not your routine breast cancer way. But I think it certainly has promise. And Andrea, like she said, what after EV Pembro? We need novel therapies which are effective, and if they're biomarker driven and well tolerated, I think. So I think, yes, this drug has a future, and we need such novel agents.
Carla, on a scale of 1 to 10, how excited are you about this drug in bladder cancer?
Yeah, I mean, I would say about a 7 or so. I think it's interesting. It's a different mechanism of action. And I mean, one of the things just to, think about SG for a moment is that, during the pandemic, one thing that we were advised to do is to try not to give chemo, which is really hard as a medical oncologist to not give chemo. But we've been using a lot of GCSF in our patients who have been getting chemo across the board of GU tumors, and we've seen significant reductions in neutropenia and all of that. So I think it's something to think about, that neutropenia in our patients is a big deal. And with SASI, I think we need to think about that even more. As we look at this drug, it has a different toxicity profile, and this is sort of what Andrea was getting to, the fact that we may be now looking at how these toxicity profiles play out in terms of deciding in a drug that has a similar efficacy, which one are you going to choose in which setting?
There are no randomized phase 3s announced that I'm aware of in bladder cancer for this. I was under the impression that you had to do a confirmatory randomized phase 3 if you had FDA approval. I've not seen that study launched yet. We'll have to wait and see.
But we are getting HER2 testing on all our patients, and we are starting to give this drug, and I'm excited about it. I've seen efficacy, so I think it's exciting drug.
And it's great to have options for the patients, and I think the good point that you mentioned is the HER2 staining has to be with the right assay. So it's a gastric cancer scoring algorithm, and now we're talking with pathologists and say, guys, can you please test
We need to work on this issue because not everyone's doing that.
Because we're not doing testing.
That's not like that. It's the Wild West out there, I tell you.
And that's a big need. So that's a major issue, the right assay for the right
We've got to keep going. We've got to keep going.